Hello everyone, welcome to this channel, Enox Tutorials. Don't forget to like and subscribe to the channel after watching so you can receive every update that we put on this channel. In this video, we are going to focus on the diabetes mellitus. That is an important metabolic disorder which occurs in a human system. So don't forget to like and subscribe after watching. If you have any question regarding the topic, just drop the question at the comment section so you can respond to it. So the objectives of this video today is going to be on, we are going to look at glucose absorption from the gastrointestinal tract at the GIT. We are going to look at the structure of the pancreas. We'll look at the biochemical causes of diabetes, that is how insulin functions in the human system. We're going to look at types of diabetes. Then basically the clinical diagnosis of diabetes. So this is where our video is going to focus on for today. So when you say diabetes mellitus, it is very simple. It's just any metabolic disorder that is characterized by chronic increase into the blood glucose level called hyperglycemia. So if a disorder leads to a chronic increase in the blood glucose level, then we call it what? Diabetes. Basically, the term for increase in the blood glucose level is called hyperglycemia, and it comes from several bases, or it can be caused by several factors. So we're going to look at what causes diabetes, the metabolic reasons or the metabolic reasons behind diabetes causes, and how we can characterize or let's say diagnose diabetes in the clinical labs. So basically, this is a GIT system, a human alimentary canal. So what happens is, after digestion, the stomach converts the food that you eat into glucose. Then this glucose, when it's being broken down, or when this glucose finds itself in the small intestines, it is being absorbed from the small intestines into the bloodstream. Now in the bloodstream, what happens is there's going to be an increased level of glucose. When there's an increased level of glucose, that glucose is now moved to the various body cells for use. So when the stomach converts food into glucose, the glucose then enters the bloodstream, that is being absorbed from the small intestines to the bloodstream. Now in the bloodstream, the body must find its way to clear off those glucose from the blood. So an organ called pancreas produces a hormone called insulin. And that insulin is what enables the body to clear over the glucose from the blood. So some of this glucose may enter the cells for use. Some of this may be stored in the liver as glycogen. So this is the stomach. The intestines, basically when the food leaves the GIT, the food enters the blood system, the hepatic portal system, as shown here. So in the bloodstream, an insulin produced by the pancreas enables the glucose increase level in the blood to clear off. So the glucose in the blood can now be cleared as what? For use in the muscle, or it can be stored in the liver as glycogen. So in the muscle, it undergoes what they call glycolysis to produce energy in the form of ATP. In the liver, it undergoes what we call glycogenesis to synthesize glycogen from what? The glucose, from the glucose. All these two mechanisms try to lower the glucose level in the blood. So this process occurs in response to what? Production of insulin. Now let's look at the structure of the pancreas proper that aids in production of insulin. Basically, the pancreas is an accessory organ of the digestive system. It is just positioned in between the liver, just below the liver, in between the GIT, that is the canal, and the spleen. It has several functions, of which enzyme production is the most important. Pancreas produces pancreatic lipase and amylase for the production of or for breakdown of fats into fatty acids, for breakdown of lipids into fatty acids, and also to break down of carbohydrates into what glucose. So pancreas produces what pancreatic juice, and that juice produces multiple of enzymes that function to break down carbohydrates into its simpler unit. Additionally, it contains what we call an isolate. Shown here are the isolates of the pancreas. This isolate plays no role in the production of enzymes. Yeah. So these isolates are cells found inside the pancreas that comes in two forms. So the isolate comes in two forms. The cells of the isolate can either be alpha cells or beta cells. So you say you have two main types of cells found in the isolate of pancreas. So we have the alpha pancreatic cells and the beta pancreatic cells. These two cells are very important when it comes to regulation of glucose. These two cells are what produces what we call hormones that produces or that aids in glucose metabolism. The hormones produced by the pancreas or the isolate of the pancreas is insulin and glucagon. So the insulin is basically produced by the beta pancreatic cell. So the beta pancreatic cell produces insulin, whereas the alpha pancreatic cells produces what we call glucagon. Now the insulin is produced in response to what high glucose level in the blood. So whenever there is high glucose level in the blood, the body produces insulin that helps to clear all the insulin or all the glucose from the blood. Additionally, when there is low glucose level in the blood, the body produces what? Glucagon that enables the blood to increase in glucose level. So we say insulin is produced in response to hyperglycemia, whereas glucagon is produced in response to hypoglycemia. So these two hormones, this insulin hormone is the most important hormone that come to play or to produce or to clear off all the glucose from the blood. That means if the insulin is deficient or is not produced in the blood or by the liver, all the glucose are going to be stayed in the blood without being cleared. So the basis of diabetes all goes around insulin. So any factor that may hinder insulin production can be a biochemical cause of diabetes. So insulin is the most important compound or hormone found in the human system that its deficiency or its inadequacy can lead to diabetes. Now let's look at something. The main role of insulin in the human system is it enables for the uptake of glucose by the tissues. It enables glucose utilization in the human system. It has a hypoglycemic effect, so that it means that it helps to bring down the blood glucose level. This one, it helps the cells to make use of the glucose being absorbed. Now it helps the body to uptake glucose from the blood, for the tissues to uptake glucose from the blood, and also it helps glucose be absorbed from the small intestines. Other functions include it helps the body to synthesize lipids called lipogenesis. It has an anti-lipolytic effect, that is, it prevents breakdown of lipids. It prevents synthesis of ketone bodies called anti-ketogenic effects, and other general effects which is not really related to the topic. So all these mechanisms of insulin at the end of the day helps to clear all the blood glucose from so that it can be reduced. That means that if this insulin is hindered, what is going to happen is all these functions are going to halt, and the glucose level in the blood is going to increase. That when it consists or when it persists for a while, it can lead to diabetes. If it is just a one-time increase in the blood glucose level, it's called hyperglycemia. That is not dangerous. But if it is a disease that relates to insulin production disorder, then we say it was a diabetes. So how does the insulin work? So let's see how insulin works to clear off what? Glucose. So let's say these pictures shown here are the cells. Shown here are the pictures of the cells. Now on the cell surfaces are what we call glucose channels. The channels are transporters that opens and close to allow glucose to enter the cells. Additionally, there are protein compounds found on the surface of the cell membranes of each cell that we say they are receptors to insulin. They are receptors because they are having a loci that can be recognized by insulin. What the body does is, whenever insulin binds those receptors, signal transductions are sent to the glucose channel for it to open. So if this insulin binds the receptors on the surface of the cells, signals are being sent to the channels found on the surface of the cell to open. That channel is called glucose transport 4 in some other tissues. So when insulin binds the receptors on the surface, signals are sent to the transporters to open so that the glucose can enter. So here is the initial step. When the insulin binds the receptors, that's where the channel opens and all the glucose will enter the cell. So here we assume these glucose are found in the blood and these ones are in the tissues or the cells. So upon the binding of the insulin, the glucose can easily enter the cells because the transporters or the channels are opened. This means that if this insulin is unavailable, this channel keep open and the glucose can never enter. Additionally, if these receptors are present on the surface but can't be sensed by the insulin, it means that glucose, or let's say insulin, can't bind the receptors, hence glucose can still accumulate in the blood and increase concentration. This condition is what causes diabetes. So when insulin is this deficit, it can lead to diabetes. So basically, the insulin we say it unlocks the channel for glucose. When it binds the receptors, it opens the channels, then glucose will enter the cells. Now let's see the types of diabetes that we have. So we have two main types of diabetes, but some other diabetes also some recognized. The main type of diabetes are what we call type 1 and type 2. These types all go around insulin. So the type 1 diabetes is an autoimmune disease, whereas type 2 diabetes occurs when there is an insulin resistance. So let's focus on type 1 diabetes. For the type 1 diabetes, what happens is, as we said, it is an autoimmune disease caused when the body's immune system tries or let's say fights the beta cells of the pancreas. So when there's an autoimmune damage to the pancreatic cells, the beta pancreatic cells, the body cannot produce insulin, or it can produce insulin, but it will not be enough to clear the glucose. So when there is an autoimmune damage to the beta cells of the pancreas, it can lead to little or no insulin produced. That means that there is going to be no signaling of the receptors to open the channel. Hence, glucose will still accumulate in the blood. Type 1 diabetes is an autoimmune damage to the beta pancreatic cell that will block production of insulin or lead to production of insulin. That means that when there's a damage to the beta pancreatic cells, insulin is not produced. Hence, there is not going to be signaling of the cells of the pancreas, or let's say signaling of the glucose channel to open. Hence, glucose will still accumulate in the blood. This is type 1 diabetes caused by what? Autoimmune damage to the beta pancreatic cells. The next one, called type 2 diabetes, is not really autoimmune damage, but there the insulin is produced, alright. For the type 2, we said insulin is produced at the end. But what happens is there is an insulin resistance. The resistance can be this insulin not being able to recognize, or this insulin not being able to recognize the receptors, or even the insulin binding but not being able to send signals to the channels to open. So for the type 1, the insulin is not available, or it may be available but not enough. But for the type 2, the insulin is available alright, but is not able to sense the receptors. This is basically caused when some other compounds can bind the receptors and block the insulin from binding. Some compounds like fatty acids can bind the receptors and block the insulin from binding. So when the insulin is stopped from binding the receptors, signals can be sent to the channels to open. Hence, there's going to be accumulation of glucose in the blood because so far as the channel is locked, glucose can't leave the blood and enter the cells. So for the type 2 diabetes, it is caused by what? An insulin resistance. Either by what? It's being not be able to bind the receptors, or maybe not binding, being able to bind but not being able to send signals through the channel to open. This condition normally found in those with obesity, those with high level of fat in their system. Fat is the most important blocker, or let's say inhibitor of insulin from its receptor. Sometimes it can bind the surface of the receptors and block the receptors from being recognized by the insulin. And that is type 2 diabetes. So for the type 1, it is when there's a damage to the immune cells, to the pancreatic cells. But for the type 2, when the pancreatic cells is working alright, but there's a resistance to the insulin. Now some other types of diabetes is gestational diabetes and MODY. Now for as the name implies, when we say gestational, a period within pregnancy. So normally during pregnancy, women tend to have an increased level of glucose in their system. This is because there must be enough glucose so that the fetus can be fed with some. So that periodic increase in the glucose level is not really a disorder, but it is a condition which must occur so that the fetus in the system or in the uterus can also benefit from the glucose. So that chronic increase in the blood glucose level during pregnancy is what we term as gestational diabetes. The next one is what we call the MODY, which normally occurs in the young people, children. So the MODY stands for maturity onset diabetes of the young. Maturity onset diabetes of the young. This one normally occurs in toddlers or infants. For them, because they have an underdeveloped let's say pancreas, the glucose level always gets increased in their system because the insulin is not produced in enough quantity to clear those glucose. So this one normally occurs in the young. So let's look at the type 1 and type 2 diabetes. So as we said earlier, for the type 1 diabetes, when there is a damage to the pancreatic cells, there is less insulin produced, or even there's no insulin produced at all. So when it affects the glucose in the blood, there is still going to be increased level of glucose in the blood because there is going to be little or no uptake of glucose by the cells, or little or no uptake of the glucose by the liver for glycogen synthesis. So there's less glucose being stored or being used in the organs when there is less insulin produced or no insulin produced from the type 1 diabetes. Additionally, for the type 2 diabetes, what happens is, as we said earlier, the body is not being able to signal the insulin. So here we produce enough insulin, alright. We produce enough insulin, but the body is not being able to signal by the insulin. That is, the receptors on the cell surfaces can't signal the insulin. So we are still going to have enough insulin, all right, produced, but the glucose are still going to be found in the cells because the cells can signal the insulin for glucose absorption. Just less may be absorbed, all right, but not enough. If the rate of glucose absorption from the stomach is higher than the rate of utilization by the cells, there's going to be an increased level of glucose in the blood. And that can lead to diabetes being occurring. So the pancreas produces insulin, but the body can't use it efficiently. Why? Because the cells have receptors that can be signaled by the insulin. Now let's try to compare and contrast the type 1 and type 2 diabetes. So for the type 1 diabetes, because it is an autoimmune disease, it cannot be prevented because it will occur, but it can be managed. Because for the type 1 diabetes, here we assume there's little or no insulin produced, so you can undergo a cutaneous injection of insulin to cure diabetes. Now for type 2 diabetes, this can be prevented through a lifestyle. So let's say when there is enough fat in the system, you can have regular exercise to melt the fat, and that can clear off the fat from the surface of the receptors so the insulin can bind. So here the body does not create enough insulin, yes. Just like this one, the body may not be able to create enough insulin. Here the causes are basically unknown and they may be genetic. So it is an autoimmune disease where the body itself tries to kill the beta pancreatic cell. So the causes may be genetic because it is unknown. This one requires insulin injections for life for the type 1 diabetes. This one, the causes may be genetic, aging, inactivity, or maybe obesity. So this one requires insulin, has needed injected, or it can be taken orally. Each of them may cause other serious health problem. They all require healthy lifestyle and medical supervision. And each diabetes have symptoms of chest, frequent urination, and odd blurred vision. Now let's look at the clinical diagnosis of hyperglycemia in the hospitals. So normally, the medical tool we use for detecting glucose in the human system is called glucometer. That tries to sense the amount of glucose found in the blood. So you have several clinical diagnostic tests which can be used to detect glucose in the blood. Each of which tries to employ the glucometer to measure hyperglycemia. So you have what we call A1C test or HbA1c test called the glycated hemoglobin test. This test tries to measure the average amount of glucose in the human system for a period of, let's say, 3 months. Basically, we believe that if the person is having diabetes, at some point, the hemoglobin is going to be glycated, that is, it's going to be fused with glucose. So if we measure, let's say, the glucose level in the blood for a period of 3 months, and the average is basically the same, there's no reduction, then we can see what the person is having diabetes. Glycated hemoglobin test. The next one is what we call the fasting blood sugar test or fasting blood glucose test, FBS. So here, this one is carried out by allowing the person to fast for a while, then detecting the amount of glucose, then you do it again. So even if the person fasts and the amount of glucose is still the same, then we can deduce that yes, the person is having what? Diabetes. The next one is random blood sugar test or random blood glucose test. So here we just pick the blood at any point in time. So we do it randomly. So randomly pick whether the person has eaten or not to detect the amount of glucose in the blood. If it is still the same, then we can say what? The person suffers from diabetes. Or if, let's say, when the person fasts, the glucose level is the same, then realize when the person eats, it increases. And if it is above the normal, then you can say the person suffers from diabetes. The last one we can talk of is what we call that oral glucose tolerance test, OGTT. So for here, what happens is we normally allow the person to ingest glucose. So the person just eats glucose. Then when the person eats glucose orally, we take the person's blood samples and measure it for some period of hours. Let's say 1 hour, 2 hour, 2 hours, and 3 hours. So we believe that if the person ingests glucose after 1 hour, 2 hour, 3 hour, the level of glucose must decrease. But if the person takes glucose orally, then realize that the amount of glucose found in the system for 1 hour, 2 hour, and 3 hours is the same, then we can say what the person is suffering from diabetes. So these are the basic clinical diagnostic tests for what? Diabetes detection. Others may be present, but the basic ones are these which are covered in the video. So the most common curing methods for diabetes is, as we said earlier, is just by injection of insulin. So if insulin is injected, it can help to cure diabetes. You can have regular exercises to cure type 2 diabetes, or some commonly synthesized drug that hinders amylase from breaking down starch into glucose called acarbose can be taken to minimize or manage diabetes. Thanks for watching. Don't forget to like and subscribe to watching so you can receive every bit of updates from this channel. Thank you.